We are going to finish looking at the B lymphocyte development, and then we're going to go here for the lymphocyte activation. All right, so we have looked at how the B cell is developing in the bone marrow, right? So we see that first you have to do the rearrangement for the heavy chain, and then you have to test the heavy chain, right? So you're going to test the heavy chain using that surrogate light chain, right? So the surrogate light chain is already a protein that is encoded by a gene that everybody has. That gene does not do rearrangement, it's just expressed whenever it needs to be expressed, okay? And the surrogate light chain is basically made of two stretches of polypeptides, right? So you have here the V pre-B and the lambda 5, right? Those are the two peptides that make up your surrogate light chain, okay? And the use for the surrogate light chains, although I don't have the picture here, is that when they are able to interact with each other. So surrogate light chains are going to interact with each other, and they are going to bring close together the heavy chains of two or more B cell receptors. Okay, so these surrogate light chains are going to interact with other surrogate light chains, and they're going to bring these pre-B cell receptors in close proximity. Okay, so the pre-B cell receptors, what do they have? They have the heavy chain that they are testing, they have the surrogate light chain, and they also have these other two immunoglobulins. So those are very, very important for signal transduction, right? So this is your Ig alpha and Ig beta. Now, once you start making Ig alpha and Ig beta, you don't stop making them. So the B cell is going to start expressing them, and it's going to keep on expressing them until the rest of its life. Okay, it expresses them forever. Right? So when you look at this whole complex of proteins here, this is what you call your pre-B cell receptor. Okay? After the heavy chain is found to work properly, so when you have a good heavy chain, so this is when your B cell is going to start to proliferate. Okay, so it's going to start dividing and dividing because it just went through a big milestone in its development. So it made a successful heavy chain. So now it can make a lot of clones of this stage in life in order to make all different types of light chains that are going to be tested later. Okay, so at this stage here, after heavy chain gets a the checkpoint, the check mark, then the B cell starts to divide a lot. Okay, so this goes to the largest proliferation stage. All right, so where is that happening here? This here was when you finally got the, right here, right? When you get the checkpoint for your B cell, for your heavy chain on the B cell. Okay, heavy chain works fine, so this is your first checkpoint, right? So then what's going to happen? You're going to start upregulating your light chain, right? Then you're going to go and test your light chain, okay? Remember that for the light chain, you have two types of genes that you inherit for both parents. So you have the kappa and the lambda, right? So what is happening here? At the light chain, after you make the light chain, now you're going to test the receptor to see if it is going to be activated by self-antigen. Remember, this is your negative selection, okay? So now a couple of things are happening here. First thing that's going to happen is that when you make your light chain, you are going to also undergo that business of allelic exclusion, right? That means if you make a rearrangement with your genes that you inherited from dad, and that rearrangement is successful, so now you stop every kind of rearrangement of the gene from mom. Okay, so that is allelic exclusion. Now, besides allelic exclusion, the light chain has another mechanism that goes on at this time, and that is called isotypic exclusion. Okay, so that means you are going to use only one gene, one type of gene for the light chain from either mom or dad. Okay, so let's say your light chain from mom worked, but it's not any of the two light chains. It's going to be the kappa light chain first. The lambda is going to be quiet. Okay, so let's say now the kappa light chain from your mom worked fine. Okay, fine. So you're going to test your B cell receptor. Let's say your kappa mom light chain did not have a successful rearrangement. So now you go to the kappa light chain of dad. Okay? You don't do lambda from mom. No, you go for kappa of dad. So now let's say kappa of dad did not do a good job. So now you go for one of the two lambda that are left. Okay? So first you're going to do your allelic exclusion with isotypic exclusion that is going to favor kappa. If kappa fails, then you go for allelic exclusion of your lambda. Okay, so first use out the kappa. If they don't work, then you go for the lambdas. So at this stage, the B cell receptor has 4 chances of getting the light chain correct. Okay, and this is important because of the mechanism also that you are seeing here. Because the whole thing, of course, you're going to test whether this thing is self-reactive or not, right? So this is very important for central tolerance, right? It's happening in the bone marrow, so it says the central tolerance, okay? Another interesting here is that after you make your receptor using the kappa, right? If the receptor works, that's fine. Then the cell gets a signal to leave the bone marrow. But then if the receptor doesn't work, this is when your B cell undergoes this rearrangement of your receptor. So this is called receptor editing, okay? Because at that stage, when you already have your B cell receptor, so you're going to test your B cell receptor, right? So you had already downregulated your RAG genes, your light recombination genes. But if your receptor didn't work at first, now you're going to upregulate again those rearrangement genes to try to get the receptor right using the lambda genes. Okay, so all this process of okay, it didn't work. Let's get to rearrange the receptor. So that's the receptor editing part of your B cell receptor development, and everything here is happening in the bone marrow. So this is why it's called central tolerance. Okay. All right. So let's see what else is happening over here. So here I put some words for you to review, okay, and understand more what's going on, because I know a lot of you are not reading the book, and I totally understand why, because I was trying to read the book to prepare for this class, and I got totally confused, and the amount of information there is like, oh my God, okay, I was getting overwhelmed, so I totally get it. So here's a more in detail explanation of what you need to know, okay. So here I just mentioned this, okay. And remember, what are the genes involved in your rearrangement, right? And when you have the joining of the two genes, what is going on with addition of nucleotides? Remember, you have the P addition and N addition. So P is for palindromic, N is for N-terminal addition, right? Of your nucleotides. Remember that the N-terminal addition is only going to happen at the heavy chain, where you have a difference between adding those nucleotides between the heavy chain and the light chain. Okay. Okay. So here you have your surrogate light chain, a kappa chain, and blah, blah, blah. Okay, what is important here? Okay, here is what's important. Let's say here that your cell is developing from the pro-B stage, right? That is when you have your pre-B cell receptor to the pre-B stage, right? And in order for the cell to keep on developing, you need to give it certain survival signals, okay? So you have a certain transmembrane protein that is a tyrosine kinase, okay? That is very important to keep to be working for the B cell to progress and to mature. Okay, so what is happening over here? You have to have this protein on the surface of the B cell, and you need to keep expressing this protein in order for your pro-B cell to become a pre-B cell and to keep on maturing. If you do not have that protein on the surface of the B cell, the cell is not going to have enough stimulus to stay alive. So that is going to be a signal for the B cell that it should die. Okay, so this specific protein for the B cell survival is called Bruton's kinase. Okay, Bruton's tyrosine kinase, or is a BTK. Okay, the gene for this protein is found in the X chromosome. Okay, so most of you took genetics, right? If you have a gene that is found in the X chromosome, what gender are you expecting to be more affected by a problem in the gene? All right, let's go for the vote. How many people think man? Okay, how many people think woman? One person. Okay, so it is the man. Why? You have only one X chromosome. You get the bad one, you're doomed. With the women, you got two X chromosomes. If one doesn't work, you get a backup one, right? But with the men, whatever they get in the X chromosome, that's all they got, okay? So now you see here, if the man inherits a bad gene for the Bruton's kinase, okay, then what's going to happen? What is this gene? Why is that protein important? Is working something on development of the B cells, right? And what is it that is doing? It's sending some type of survival signal. Okay, it's going to tell the B cell that it should stay alive. B cells need a lot of survival signals, because if they're not kind of stimulated, they'll become like zombies and they'll end up dying. So they need survival signals. Now, if you inherit a bad Bruton's kinase and you don't give a survival signal to the B cell, what's going to happen to the B cell? It's going to die. Are you going to have any B cells around? No, that's a big problem. Okay, so for the people who inherit this disease that is X-linked, the disease is called agammaglobulinemia. That means they do not have gamma globulins. The prefix A something means missing something. So here is telling you that you don't really have antibodies, or you don't have the B cells to fight whatever the B cells fight, right? And what is the B cell most important for? Production of antibodies, and is a part of your adaptive immune system, right? So these people have big problems, with lack of B cells, of functioning B cells, okay? So basically, he's telling you that if you inherit XLA, you don't really have B cell maturation, okay? What are the B cells fighting in the body? Like, what is the main function of the B cells? producing antibodies, and they can also recognize what kind of pathogens. What kind of pathogens can B cell recognize? How do they recognize pathogens? 3D. 3D. So it has to be a pathogen that is where? Where are you going to find a pathogen? Okay. B cells are going around, right? Where is the B cell in the first place? Yeah, there are secondary lymphoid tissues, but they are in the contact with lymph, right? So they're over there, and they are waiting for pathogens to show up, right? And then they're going to recognize the pathogens by 3D, right? In order to do that, where are the pathogens? In the blood. Okay, you said in the blood. It could be in the blood, but in this case, the B cell is sitting in the lymph node. But it's where? You're getting your lymphocytes crossed. Okay, how is it that they're going to present? How is it the B cell is going to see the pathogen? Who's going to present it to them? Antigen binding. Which ones? Yes. The dendritic cells or some antigen presenting cells, but the dendritic cells are special because they're going to bring a pathogen to be presented to the T cell, but without chopping up the pathogen. They're going to bring it like they're handing the whole pathogen to the B cell. The whole thing, the whole, let's say, the whole bacterium that it found. Okay, so it finds a pathogen, it brings to the B cell, and you say, here, this is the whole thing. And then the B cell goes there and say, oh, I feel the 3D of this pathogen. And that means it's a pathogen that is found outside, in the outside environment. It was an extracellular pathogen. Okay? So the pathogen that is floating around goes in there, is presented to the B cell. So now the B cell can start fighting that pathogen that is extracellular. And if I say extracellular pathogen, what is the first kind of pathogen that comes to your mind? Bacteria, right? So if you have a person that has XLA, what kind of infections that is most likely going to have that is recurrent infection? Extracellular bacterial infections, right? That would make sense on how the problem with this gene can affect a type of reaction to certain types of pathogens. All right, so let's see here in more details what is happening with your B cell throughout the development, right? So remember, it is arising from this multipotent stem cell, and everything is happening here in the bone marrow. So you have this stromal cell that is like all over the bone marrow here, okay? And the stromal cell is presenting certain ligands to receptors on your developing cell, right? And the presence of those interactions, and also presence of certain cytokines that are produced by your stromal cells, those are the signals that are going to tell your B cell to become a B cell and differentiate. And we have seen some of those before, right? So first you have here your receptor tyrosine kinase, general receptor for lymphocytes, right? And it interacts here with your stromal cell, right? Your stromal cell is going to produce a certain cytokine that is going to stimulate the hanging around of those lymphocytes in the bone marrow. Okay, so here you have certain cytokines that are getting produced. Now, at the same time here, you have these very important cytokines that are getting expressed here, that is the interleukin-7. Okay, so interleukin-7 is very, very important for committing the developing cell towards the lymphoid lineage, right? So once you get activation of this lymphoid lineage, this is also a survival signal, okay? So let me see what is important here. Okay. I don't want to get too much information here. Okay, so you do have the interleukin-7 receptor on this common lymphoid progenitor cell, right? Now, up to this point, this common progenitor could become a T cell if it wanted to. But then you have certain things that happen that are going to really shift the development towards your B cell. And here is this is a very important stage in the B cell commitment. Okay, so you're going to have your receptor tyrosine kinase that is going to be start to get expressed on your B cell. Okay, so this receptor tyrosine kinase is also called CD117. Okay, so we go by the short name of this thing that is called KIT. Okay, so it is going to bind to a ligand on your stromal cell that is called stem cell factor. Okay, so stem cell factor binds to KIT. Okay, once you have this interaction happening, the cell is going to be committed to becoming a B cell. So after the expression of KIT, you are going to start expressing those B cell specific transcription factors that we briefly had seen before. So that was the early pro-B cell transcription factor. Okay, so here you're going to see right there, early pro-B cell transcription factor. Okay, and this is your E2A right there. Okay, and then you're going to have the PAX5. That's what also we have seen before. So E2A and PAX5, those are the transcription factors. They're happening inside of the cell. Okay, so they're being expressed over here. They go to the nucleus. Those are the ones that really commit the progenitor towards a B cell lineage. Okay, so after that, you're going to have your whole development of your B cell receptor, right? So after you express EA2 and PAX5, you're going to start expressing your heavy chain, your light chain. You go from the pro-B to the pre-B. Remember, when you shift from a pro-B to the pre-B, at the pre-B, you get your B cell receptor, right, with surrogate light chain. At the pro-B, you don't have receptors on the surface. Okay, so here's like the whole development going on, right? So you go from no receptor to commitment to still, pre-B receptor, there's goes here, and then to full maturation. So important things to know here are that the first thing for commitment of your B cell is expression of the KIT. And I like to put a picture here of your kitty cat, right? So that's the little thing that's going to grow, right? So express KIT, you're going to be a B cell. Like what kind of B cell? You're going to be transforming that B cell into a beast with M spikes. That's your IgM receptor on the surface. Okay, so you grow from the kitty cat to the beast with IgM on the surface. Okay. Important things to know about your transcription factors. Remember, Icarus is important for lymphoid lineage, so B cells and T cells are going to have it. But then here, your E2A transcription factor is really the one that is important for commitment of your B cell. Okay, E2A and the PAX5. E2A actually goes expressed a little bit earlier than your PAX5. But once you express those two receptors, so this is when you have a total commitment for B lineage. Okay. All right. So what are the genes that are important that are going to do the rearrangement? So in comparison to what we have seen before with the T cell, right? So you have here your rearrangements genes, RAG1 and RAG2, right? Remember that is important for formation of your heavy chain, and that they stop expressing for a while, right? And then you're going to get your light chain rearrangements, and then they stop being expressed, right? And remember that also when you're doing your heavy chain, you have that TDT expression, but that is not going to happen for your light chain. This should be a little bit more to this side here. Okay, light chain is not going to express TDT. Okay. And here the genes that are important for growth, maintenance, and B cell characteristics, right? So we just mentioned here the BTK, the Brutus kinase, that is important for signal transduction, right? So when is the BTK coming into action here? Here we see at the pro-B stage. Okay, so pro-B stage before the pre-B, and then it keeps on getting expressed. It doesn't stop, okay? Same story here goes with your Ig alpha and Ig beta. Those are always going to be needed for activation further down the line of your B cell. So once they get expressed, which is early, right, early in this pro-B stage, they never stop the expression. They keep on expressing for the whole life of the cell. Okay, so this is important. Now I'm getting this program is not working signal. So this is important, right? And we found those two here. Once they get expressed, they never stop expressing, and this is all for signal transduction, right? And we have seen here also for the KIT, right? KIT is important only in the beginning, right? For this early pro-B stage, so only for your pro-B that you're going to see expression of your KIT, right? And here we also see your interleukin-7 receptor that is important up to your pre-B stage. So pro-B to pre-B, remember interleukin-7 is also important for your T cells, right? So here is important for the beginning. Once you get into your mature pre-B stage, then you don't express this interleukin-7 receptor anymore, okay? Another thing that is important here that I didn't mention before is this guy over here, CD19. So let's see when CD19 starts to get expressed. Like right here, look at this. Pro-B stage, and then it doesn't stop anymore. Okay? CD19 is a very important surface marker that is characteristic of B cell. So all B cells have CD19. Okay? Same way all B cells have BTK, but CD19 is like one of the classic markers for B cell identity. Okay? So let's see here the ones that are only expressed temporarily. Of course, you have your surrogate light chain. They only express when they needed. You have your pre-B cell receptor, right? And then you have here your KIT and interleukin-7, right? And then you look at the ones that are important for signal transduction, signaling, that once they start getting expressed, they never stop. Okay, so this was the idea for your immunoglobulin alpha and beta, BTK, and CD19. Those are always needed for cell function. Okay, so then what happens here? Once you have your functional IgM on the surface, remember mu is the first one that gets expressed. Once you have it expressed, the cell is actually ready to leave the bone marrow. And this is the part that I had to do some other reading now, because I had learned that in the bone marrow, you also express the other isotype of the first B cell receptor, the IgD, okay? And now I read on the new edition of the book that that's not really what happens. And this is the story with immunology. We keep on learning on immunological mechanisms. So what happens here is that the B cell, once it has IgM, the B cell is going to get signals that it can leave the bone marrow and go to the periphery. So the B cell leaves and it goes to the spleen. At the spleen is going to undergo another round of testing to see if it is going to see antigens that are part of your body but are not expressed in the bone marrow. Okay, so in the spleen is going to undergo another round of negative selection. And if it passes, then it is going to express the IgD. Okay? And then it can go to other stages. Okay. All right, so here is when I put this slide here. The B cells are leaving the bone marrow when they have only IgM on their surface. Okay, so the IgM are positive. Then what's going to happen? Remember that those B cells, once they made the IgM, they tested. If they passed the test, that's fine. They passed negative selection. But then if they did not pass the test, that's when they undergo receptor editing. Now let's say that now they passed the test, they made the light chain. What's going to happen here? Look what's going to happen. They are going to upregulate a form of a protein on the surface that is actually a G protein coupled receptor. Okay, so this protein is called S1PR1. This protein is going to bind a ligand that is present in the blood. Okay, so as this cell undergoes negative selection and it survives, it starts to upregulate the receptor. So the cell here, okay, is in the bone marrow. It has a proper B cell receptor. It passed selection, so now it starts to make this receptor here that is called the S1PR1. And as time goes on, it gets more expression of this receptor and more expression of this receptor. So now it can feel, this is inside the bone, okay, this is all inside of your bone. So we put a bone over here. This is our bone, okay. Now it can start to feel the presence of these ligands that are in the blood, okay. It is going to have high affinity for those ligands that are in the blood. Do you have blood in your bone? Oh yeah, the bone is highly vascularized. So you have this thing that is coming here in blood vessels, okay? And this is going to call on this receptor because you're going to start binding to it. And the more receptors you have, the more binding you're going to get to the S1P that is in the blood. So that is going to give a signal for your B cell to leave your bone marrow and go to the circulation. Okay? So it leaves the bone marrow, goes to the circulation, and then it's going to have other signal that is going to make it land inside of the spleen. Okay? When it reaches the spleen, it's going to stay there for further developing. Okay? So this is the part that I was missing before. All right. So at this stage here, it leaves the bone marrow, it goes to the periphery, it goes to the spleen, and then in the spleen is going to be tested again with other self-antigens to see if it passes the second stage of negative selection. So let's say that it passes, so now at this point it is going to start to make IgD. Okay, it's going to make IgD and IgM, two of them on the surface, and it's going to be allowed to recirculate to the rest of the body. Okay, now it can go to the lymph nodes, and it can keep on recirculating. But it doesn't do as much recirculation as you're going to see with T cells. T cells really like to go around a lot. B cells do recirculate, but not as much. All right, so here, one question. And this is like clinically relevant, right? Okay, let's see here. If you have a problem with your surrogate light chain, what is going to be the overall problem of your immune system? So your surrogate light chain doesn't work. So now you have to think, when is it that you're expressing, what is it that you are testing, and what is going to happen to this cell if your heavy chain seems like the heavy chain doesn't work, right? So heavy chain doesn't work because your surrogate light chain is not working. What's going to happen to your cell? What's going to happen to the body of the person that has the cell? Does anyone need more time? Everybody answer? Yeah, not working? Oh, got issues with internet today. Okay, I'll see after the class if I can put you there. Okay, what's your answer? It is B. Okay? You are not going to be producing those B cells. So you're going to have recurrent bacterial infections because those are your extracellular types of pathogens. Okay? Somebody answer here, excessive B cell proliferation. Not really, because this is the first checkpoint to see if the cell is going to be allowed to survive or not. If your heavy chain is found to be non-functional because your surrogate light chain is testing it, if your heavy chain is non-functional, the cell is going to try with the other gene and then it's going to die. It's going to undergo apoptosis. What would cause excessive proliferation? Some type of lymphoma, cancer. Yeah. Yeah. Okay. All right, so the B cell, it underwent all of those checkpoints, and now it has the B cell receptor on the surface. And now let's say that the B cell has found the antigen, right, that it needs to react to, and it processed a piece of the antigen, because remember, B cells are capable of phagocytosis, right? So it processes the antigen, it gets a piece of the antigen, and it puts on its MHC class 2, right, because the antigen was extracellular, puts on MHC class 2 to present to a T cell. And now your T cell that recognizes that little piece of protein, that T cell shows up and is going to recognize it and is going to start talking to the B cell to tell the B cell to produce certain specific types of antibodies. Okay? So the T cells have a very important signal on their surface that is called CD40. Okay? This signal is a ligand of the receptor that is on the B cell, and that thing is called CD40 receptor. Okay? So the T cell has this very important job. When it interacts with the B cell, it is going to interact here at the B cell receptor, but it's also going to interact at the CD40 to CD40 receptor close to the B cell receptor. This interaction here is going to send a signal to the B cell to produce a specific type of antibodies. Mainly, it's going to tell the B cell to do the class switching of the antibodies. Because remember, up to this stage, the B cell can only produce IgM. IgM is there. This guy over here. But unless it gets a signal from the T cell through this interaction over here, unless the T cell tells the B cell to do class switching of those antibodies, the B cell is going to keep on producing IgM. It's not going to produce any of the other isotypes of antibodies. Okay, so this is a very important activation of your B cell that depends on your T cell helping it to do the job. Okay, so this is like down the line after the B cell survived and after it has seen antigen. You will need T cell to do class switching. Okay. So how is it that the B cell can do the class switching? Okay, so here we look at the IgM gene. Remember, this IgM gene of your B cell was already rearranged, and everything is really, you know, happening at the mature cell. Okay, so now this cell is not naive anymore. Okay, it's seen antigen, and now it is getting activated by a helper T cell. Okay, it interacted with CD40 ligand. It also had some cytokines to help it to decide on what to become. Okay, and here is the DNA of your heavy chain. Because remember, your heavy chain is what is stuck on the membrane and is going to dictate what class of antibody the B cell is going to produce. Okay, so what happens here is that when you get CD40 activation, the cell can shift the expression from mu, okay, from mu constant region to some other constant regions like gamma, epsilon, or alpha is missing over here, right? And you see here the switch is going to go from mu to one of those. It's going to go to the constant region, okay? So this is the type of loop that the DNA is going to do, and whatever is in the middle gets cut out of the genome, and then your B cell is going to start to make this isotype of antibody. Okay, so in this case here, it has switched from mu, which is right here. It switched from mu to epsilon. So here you're going to produce IgE antibody because it went here from mu to epsilon. So no more mu. Now you're going to start with your epsilon gene to produce your antibody. All this piece of the DNA gets cut out, is lost. So when you do class switching of antibodies, you can only go from M to something else, but you can never go back to M. Because once you do the class switching, all the middle part of the DNA gets lost, gets cut out of the genome. Okay? Everybody got this part? Okay? All right. So here, what are the different classes of the antibodies that you can produce? Right? You start with M. That's your first one. And based on CD40 activation and also cytokines, you can produce IgGs or IgEs or IgAs. Is there one type of antibody missing here? Which one? IgD is not there. Why?
Because it's on the naive cell.
Because it's part of your mature naive cell, and it gets co-expressed with your IgM. Remember, to express IgD, you do the mRNA type of shuffling, okay? So that does not depend on cutting a piece of your DNA out. So here, when you're expressing IgM, you also have IgD over there, okay?
Is IgD always present, or does it?
Not always. When the cell gets activated and you start to make this soluble IgM, I don't think you express much of the IgD. And also there is a little bit of IgD that gets secreted, but then we don't know why. Those are the parts that are still very blurry. Yeah. Yeah, IgD is, one of those black boxes. We don't know that. Okay. All right. So how do you do class switching? You go from IgM to one of these other three classes. When you talk about IgM to IgD, that's not class switching. What do you call the process of the mRNA? It escaped my mind now. Is mRNA
Transcription.
no, when you do switching from exons to different exon?
Oh, splicing.
Splicing, yeah, you see, I had just one blank with the splicing. mRNA splicing, very, very important. Am I going to ask you about that in the test? Yes. Okay.
So what was it that you said?
What is the question? Now everybody woke up. Okay. How do you get IgM and IgD being expressed on the surface of a B cell? It's not by shuffling the DNA, it's by shuffling the messenger RNA. And you call that messenger RNA differential splicing or just splicing. Okay? All right. And then you do class switching by cutting a piece of the DNA out. That's the real class switching. All right. All right, so finally we get to the lymphocyte migration and activation. All right. So what we're going to look at over here. We're going to look at the very, very important jobs of dendritic cells. Then we're going to look at how those lymphocytes are going to go from the blood to the lymph and then back to the blood, and it's going to be surveilling the whole body looking for bad guys. Okay, so circulation. Where is circulation? Then we're going to look at some chemokines that help to attract those lymphocytes, right? And those also go hand in hand with upregulation of some adhesion molecules, and we had briefly seen those before. Okay. Then finally, we're going to look at how is it that your lymphocytes know where they have to go. Because remember, they're circulating all over the body. But if you have an infection like on your hand, you really need those lymphocytes to go to your hand to fight the infection. You don't want them in the rest of the body. They can circulate because they have to hop in the blood and get to different parts of the body, but they have to come out at the site of the infection. Because if they keep on circulating, they're just useless. They have to go and fight the enemy where the enemy is, right? All right. So ideas here. First thing that we have to think about is the presence of your interdigital or resident dendritic cells. Okay, you're going to look at your tissue resident dendritic cells. That means they're sitting in the tissue and they're not going anywhere. They're just sitting there, waiting for pathogen to show up. When they're just sitting there, they are hungry. Okay, so they're sitting there waiting for pathogen. If the pathogen shows up, the dendritic cell is going to eat it. That means it's sitting there and it's hungry and is also called phagocytic. Okay, so now let's say that it ate the pathogen, is going to process the pathogen, is going to get the antigen on MHC molecule. When it does that processing, it is going to change personalities. So first it was just sitting there and kind of hungry and didn't feel like doing anything. Do you want to go out? No, I'm hungry. What about traveling in the blood? Forget about it. Oh, now I ate. I got the energy. I want to change my personality. I'm not going to eat anymore because I just ate. So it becomes non-phagocytic. Okay? And now it got energy. I want to go out. So it's going to come out of the tissue, it's going to get into the blood circulation, and it's going to travel towards secondary lymphoid organ. Okay? So at first it was just stationary, it wasn't doing anything, it was just hungry, and then it ate. It's like, I don't need to eat anymore, I'm not going to eat anything else, I'm just going to travel now. So then it goes to the secondary organ. Usually it's going to find a lymph node. Okay? So it's going to get to the lymph node by the vessel that connects entrance of lymph to the lymph node, and you call that your afferent vessel, afferent lymphatic vessel. Okay? So when it gets there to the lymph node, it is going to look for B cells and T cells. It's going to look for naive T cells to show up, okay? And is also going to present that antigen to naive B cells, all right? So it has two ways of presenting antigen. It's going to present in one way for your T cells, because T cells only see 2D. It's going to present another way to the B cell, all right? Now, this is really cool because the dendritic cell is going to travel to the lymph node that is closest to the place where it found the microbe. Okay? So it goes to the nearest lymph node or to the nearest secondary lymphoid tissue, and it sits there. It doesn't get out of there. It sits there and it's going to wait for the T cell to show up. T cell is traveling. It is traveling, and then when it gets to this lymph node, just because it travels through the circulation, travels both through the blood and through the lymphatics, when it gets in there, the dendritic cell is sitting there waiting for it. So it's going to present the antigen to the T cell when the T cell gets there. How is it that it's going to present? The dendritic cell, after it processes the antigen, it is going to upregulate a very, very important ligand on its surface. Okay? It upregulates this ligand that is called B7. Dendritic cells are the only antigen-presenting cells that can upregulate a lot of B7 on its surface, on the cell membrane. This is why they are called professional antigen-presenting cells. Okay? Do other cells have B7? Well, yeah. B cells have B7, but not a lot of them. Okay? And in order to activate your naive T cells, you have to have a good expression of B7, because the T cells will want to get activated by B7. Okay? So now let's see what happens here. You activate your T cells through this B7 interaction and presenting the antigen. The T cell is going to start to make clones of itself because it is most likely the only T cell that could recognize the antigen. Okay? So it's going to start making more and more of itself. It's going to make a clone. It's going to be a clonal proliferation, and it's going to come out of the lymph node. It's going to come out through the vessel that is called efferent lymphatic vessel. Okay, so afferent got there, efferent comes out, okay, and it's going to start looking for the site of that infection, okay, besides looking for the matching B cell. All right, so let's see what happens here. What's the whole story of how this is happening? How is it that your dendritic cell recognized your antigen? Well, the dendritic cell has on this surface, it has those pattern recognition receptors. And you thought you'd never have to look at them again. No, surprise, they're still there, right? So they have those pattern recognition receptors. They recognize the pathogen, they eat it up, they process the pathogen, they put a piece of that pathogen on your MHC class II. At the same time, it got a signal to upregulate expression of MHC class II because it's gonna need it, right? And at the same time it is upregulating MHC class II, it is upregulating your B7 ligand right there. Well, B7 has another name. You can call it CD80, CD86, right? But I just like to stick to B7 because it's enough acronyms that we have to remember, right? But if you see anywhere CD80 or CD86, in the back of your head you go, oh, that's B7. All right. Okay. So here is the continuation of the story, right? So here you are going to present your piece of pathogen to one lucky T cell. T cell is going to, this is naive T cell, right? Here is not showing the interaction with the B7, but yeah, you need their interaction too, remember? B7 here, B7 receptor, you need that interaction. you are going to activate this cell to proliferate. So it goes from naive T cell to a clonal selection. Okay, this clonal selection is rocking. It is active. It's going to do its job. You're going to get effector cells. Those are your effector cells. And you're also going to keep some of them as memory T cells. Those guys are going to live for a very, very, very long time. You want them around because in case you find your antigen again, you don't need to activate your naive guys again. That takes time. It takes, I don't know, about a week or so for them to become effector cells. But if you have memory cells present, they are a lot easier to activate. You don't really need B7 interaction to activate these guys here. The only thing that they will need is recognizing the antigen, okay? Because they will know that if they see that antigen again, you know, they are part of the clone, right? If they see the antigen again, they are going to be ready to work, okay? They're going to be ready to further differentiate into more effector cells. The memory cell hangs around for a very, very long time, okay? All right. So here I had focused the presentation of this as an extracellular pathogen. So this went through MHC class 2, right? So this was activation of helper T cells, right? But if you have presentation through MHC class 1, okay. If you have presentation to MHC class 1, your effector T cell is going to be a cytotoxic T cell. Right? Everybody knows why, right? MHC class 1 is going to interact with CD8. All right. Okay, so T cell divided bios. Let me see if anything is important here. Antigen, yeah, so just put into words here what they just mentioned. Okay. Another question for you. All right. Which of these does not describe a naive T cell? What's your answer? D. Okay. All right. So now this idea here on how you activate your naive cells, right? So here you have your dendritic cell, your professional antigen presenting cell that is coming with something on the MHC and is presenting to the T cell, right? But in this case here, if you're only presenting something on MHC, that is not enough to activate your T cell. It doesn't matter if it is CD8 or CD4 positive, it's not going to activate the T cell. The main reason is because if you have MHC molecule on the surface of the cell, that MHC is always going to have a piece of peptide that is being presented on it. You're never going to find empty MHC on the surface of a cell, presenting cell, okay? So if you are looking at, let's say something happened with the processing of antigen and then a piece of your own peptide was put by mistake on MHC class 2. If you present this to a T cell and you activate the T cell, now the T cell is going to start looking for your own presented peptide in order to kill it, okay? You don't want that to happen. So if a mistake happened here and you did not encounter antigen, did not encounter pathogen, and you put a piece of your own peptide on this presenting molecule, if that thing activated the T cell, that will be disaster. Remember, you're always going to have something on MHC molecule, okay? So in order to avoid that kind of disaster from happening, your T cell really needs 2 signals in order for it to be activated. Okay, this is why it is so important to have that. What is the name of the ligand over here? On the surface of the presenting cell? That binds to CD28? B7. B7. I would like for it to be called CD28 ligand. But is your B7? Okay? CD80, CD86. Oh God. Okay. So B7 is going to interact with its receptor that is called CD28. And this is very important to know. Okay? Because without this co-receptor CD28 activation, you're not going to get activation of your naive T cell. Okay? Question here is, what happens if you only get T cell receptor activation? Well, not only like nothing is going to happen, but you're going to teach your T cell to do nothing. If you get only one stimulus, you're going to become a zombie. You're going to be anergic. So this is an important mechanism of peripheral tolerance. Okay, because this is happening in the periphery. You give one signal to your T cell and it's telling it, okay, forget it, just be quiet, don't get activated. You have to tolerate this antigen because you did not get co-activation. So this is teaching your T cell to just become tolerant. Important for peripheral tolerance. The other name of this, be quiet, is allergy, be anergic. All right. So let's see here a little more on this co-stimulation of your naive T cell, right? So remember, you need to get the receptor here, CD28, on the surface of the T cell. That is going to be activated. Once you have B7 CD28 activation, you are going to send a signal to the dendritic cell that you have a good match here of your naive T cell. Now, your dendritic cell is going to start producing cytokines to teach your T cell how to behave. Okay? So now this is a third signal that is going to be in this interaction. Okay, so this is signal 1, this is signal 2, and now it's going to be signal 3. Okay? Some cytokines that are produced, for example, here it says interleukin 12, right, is really going to trigger a response on this T cell that it needs to proliferate. Okay, it needs to make a clone of itself. It starts out as only one T cell. Now it is going to produce this very important cytokine that is called interleukin 2. This cell is producing interleukin 2, but it also has receptors for interleukin 2 on its own cell surface. So it is producing the cytokine and it is interacting with the own cytokine that it produced. So this is a form of autocrine signaling. It produces the thing that stimulates itself. Okay, it's an autocrine type of signaling with interleukin 2. Interleukin 2 is very, very important to cause proliferation. So this is how you get a clone of your activated lymphocyte, okay, your activated T cell. There we go. Now it's going to become an effector T cell, clone of effector T cells. Okay. All right. Okay, so here is a little bit on that story. Okay, this is a first signal. So here you're going to get, oh, it didn't show here your second signal. Okay, it didn't show here, let's assume, okay, see, this bothered me here. So here you have your B7 right there, okay, right there. So it had activated already. So now you are going to get further signaling of receptors. Let me see. Cytokines. Okay. Interaction of your dendritic cell with your T cell. Okay. Through other receptors that are going to upregulate more messages, right? Yeah, so this interaction here, CD40 to CD40 ligand, is really more important for your B cells. Okay, that I want you to remember more in relation to B cells. Here, yeah, it's also important for dendritic cell activation of cytokine production, but I want you to remember here that those two interactions are really more important. Okay, so you get first signal, you get second signal, then you're going to get upregulation of cytokines and differentiation into effector cells of your T cells. Okay, but I see that is also pertinent to here that you have CD40 ligand interaction that is going to make your dendritic cell to produce specific types of cytokines. But yeah. So this is another effect that CD40 receptor is doing. Once your CD40 is upregulated, is interacting in the dendritic cell, you're going to express more and more of your B7 receptor. Okay, but this is just like a bit of too much detail here. Okay, so let's go here. Okay, that's a good one. What is your ligand for B7? I should say, what is the receptor for B7? Right. ADC. Okay. All right. So here, this whole idea, let's keep track of what's happening, right? You have your antigen, your professional antigen presenting cell that interact with T cell receptor. You have co-stimulation, and now you're going to have production of certain cytokines. They're going to tell your effector T cell to behave a certain way. Okay? And now you have to think, when you have your T cell activated, how is it going to behave? How is it going to leave the place where it is at and get into this blood circulation and reach this site of where the action is needed? Okay, so here's a big picture, right? So you know that your lymphocytes are going to be circulating, right? And they can get to certain organs that via the blood. Okay, so they can go in the blood circulation and get to certain organs. So let's say here, you can go to the liver, or it can go to the skin, or it can go to the kidneys, or it can go to the gut. Now, once they are in these organs, they can also get back into the circulation, but not directly into the blood circulation. They have to go through the lymphatic circulation first. Okay, so let's say if you have your lymphocyte on your skin and you found antigen, it does not go to the blood, it goes to the lymphatic circulation to get to your afferent lymphoid vessel and reach your nearest secondary lymphoid organ. Okay, so if you found a pathogen here on the skin, for example, it hops on the peripheral lymphatic circulation and it reaches the nearest lymph node. If you found a pathogen in your gut, it is going to hop to the lymphatic circulation and reach mesenteric lymph nodes. Those are the ones that are closest to the gut. So it's going to look for a secondary lymphoid organ that is nearest the site of infection. Okay? After that, when it leaves the secondary lymphoid organ, it has two ways, two paths that it can go. It can either leave at the efferent lymphatic vessel, or it can hop back into the blood circulation. Okay? Anyway, it's going to reach the blood circulation, because if it goes straight into the efferent lymphatic vessel, when it gets all the way up here in the thoracic duct, it's going to be dumped back in the venous circulation. But it also has the opportunity to hop back in the blood at this stage over here. Okay? All right, we'll get more description on how we can get in and out of those sites. Okay. So let me see here. Important thing to say. When it's also in the circulation, it can reach secondary lymphoid organs without going through the lymphatic circulation. It has another avenue. It can hop into the lymphatic organ by a special site on little veins. Okay, so some little veins that are getting close here, that are furnishing the lymphatic organ with blood, right? They have specific endothelial cells that are different from the endothelial cells that you find in other blood vessels. Those endothelial cells, they are called high endothelial venule cells. Okay, we have more pictures of that. But when your B or T cell, especially T cells, when they get over here, they can squeeze themselves between those high endothelial venules and hop in your secondary lymphoid organ. Okay? So what is happening over here? You have your blood vessel that is going to go here. It goes as arteries, right? It's going to replenish here all the nutrition and oxygen that your lymph node needs, right? When you have your capillary bed, everybody remembers what is a capillary bed and what is happening at the capillary bed? Right? You have little arteries, you have those capillaries that are part of your arteries, and then you have your little venules. They go back to the venous circulation. Now, when you get to the venules, those tiny vein, the blood vessels, this is where you're going to find these strange-looking, very tall, kind of fat-looking cells. This is the part that you call this high endothelial venule. You call it high endothelial venule because those cells are tall. So they're not the very skinny looking type of endothelial cells that you find on capillaries. Okay, those are tall cells. So at these stages here, then your lymphocyte is able to squeeze between those cells and hop inside of your lymph node. Okay. Now, what kind of cells? You have here your naive T cells. You have your basically naive T cells that gets in there to get activated. Okay, so they get over there either by going through the blood or by hopping in lymphatic circulation. Okay, so when you get here at your lymph node, this is where all the activation action is going to happen. So your T cell got there, it sat there and waited for the dendritic cell to show up and to activate it. If it doesn't get activated, it's going to get bored. It's going to hop again in circulation, and it's going to keep on recirculating until it finds a place that it gets activated. Okay. All right. So how is it exactly that it gets there? Okay, this is when you get to put some more details on those interactions. Okay. At those high endothelial venules, you have some very special sticky molecules that are expressed. Okay, so you have these sticky molecules, and you also have production of certain cytokines that are quite important for interaction and adherence. Okay, sticky molecule number 1, PNAD. Okay, so PNAD is for peripheral node adhesion or addressing molecule. Peripheral adhesion sticky molecule right here. That's your PNAD. That is going to be on your high endothelial venue, right? Okay. The high endothelial venue is going to secrete certain cytokines that are CCL19 and CCL21. They usually go together. Those are going to interact with receptors on your T cell. What kind of receptors? Oh, look at that. Those are receptors for CCL19. You call them CCR7 receptors. Okay? CCR7 is going to interact with those cytokines right there. Okay? When these interactions are happening of selecting on PNAD and CCR7 with the other signals, this is when you are going to get the signal to go through the high endothelial venue and reach your lymph node. Okay, so those are the important interactions that you should know. All right, so I'm going to stop here and we pick up on Thursday.